Forensics Lecture 6-3, Spectrophotometry. So spectrophotometry is an important analytical tool. It measures the quantity of radiation that a material absorbs as either wavelengths or as wavelengths and frequencies. Just as substances absorb light or visible light, many also absorb invisible radiations of electromagnetic spectrums. They can identify a substance by exposing it to a specific type of electromagnetic radiation to identify it. So the theory of spectrophotometry is the quantity of light that's absorbed at any frequency is actually directly proportional to the concentration of the absorbing species. This is known as Beer's law. So A equals K times C. In this case, A is the amount of light taken up by a single frequency. K is the proportionality of the content, and C is the concentration of the absorbed material. All spectrophotometry this allows spectrophotometry to be used as a quantification technique. So now we're past the fact that we identified this is a drug or there's a drug present. Now we're looking at specifically what type of drug this is and how much of it is present in the mixture. A spectrophotometer is an instrument used to measure absorption, basically. So it produces a graph or what's called an absorption spectrum, which depicts the absorption of light as wavelengths or frequency. The components of this photometer, are a radiation source, so a light source, a monochromator or frequency selector, which is basically a prism, a sample holder, so whatever we're looking at, whatever drug we're trying to identify, a detector to convert the electromagnetic radiation into an electrical signal, and then a recorder, which is going to produce the recording so that it can then be compared to a standard sample. Ultraviolet and visible spectrophotometry. When we talk about a UV or ultraviolet spectrum, it's a tool to determine a material's probable identity, but it may not provide a definitive result. When we look at the IR spectrum, which is the infrared spectrum, it is much more, or creates a much more complex pattern. And so different materials have distinctly different infrared spectra. So the results, the pattern that they create is very different between them. So each IR spectrum is equivalent to what they would consider to be a fingerprint of that substance. IR spectrophotometry is actually one of very few tests that can be considered in itself sufficient for identification. As we discussed in the last section, it's multiple tests that need to be performed to conclude the identification. This is so specific using the IR spectrum, the infrared spectrum, that it can be sufficient to identify what it is. They also use what's known as mass spectrometry. And this is a beam of high energy electrons that collide with materials and produce a positively charged ion. So positive ions will almost instantly decompose into numerous fragments, which then separate according to their masses. So under controlled conditions, there's no two substances that produce the same fragmentation pattern. So again, they can use this to identify the quantity of each, or the mass of each substance that's in that mixture. Most of the time, what they're going to use, or the forensic chemists are going to use, is called a CGMS, which is a gas chromatography and mass spectrometry together. Coupling these two allow a specific identification of a drug. So it directly connects, or there's a direct connection between the gas chromatography column and the mass spectrometer allows each component to flow into the mass spectrometer from the gas chromatography. And so the first part of separating the mixture into the components is accomplished by the gas chromatography, um, and then the fragments of each component um, uh, will enter into the mass spectrometer, and it will, as it flows through the mass spectrometer, it will produce distinct patterns. Again, this is like a fingerprint of the substance, and then they will take that unknown substance, that recording of the outcome of the GCMS, and um, compare it to a known reading. So this gives them more of that um, quantitative analysis um, in order to identify the drug, its makeup, how much of it is present in the mixture that they seized from whatever the crime scene may have been. When we collect and preserve drug evidence, the field investigator, as always, is responsible for ensuring evidence is properly packaged and labeled. When dealing with drug evidence, common sense is typically the best guide, obviously keeping in mind that the package must prevent loss of content or cross-contamination. Usually the container that the drug is seized in is sufficient to keep it packaged in. They just then need to label it. And all packages, as always, must be marked with the identification um, of the substance, the officer, time, location, so on, and then, of course, the chain of custody. This ends lecture 6-3. There's a quiz next class.